Welcome to our ninth already webinar at NEMIS Academy. My name is Alina. I'm Sales and Marketing Manager at NEMIS Technologies, and I'm super happy to see you all guys joining already our ninth webinar. Before we start, I would ask you guys to help me with a technical checkup. Just type in the chat room if you see me well, hear me well. Plus would be just enough that everything is fine on our way. Okay, someone is typing. Yeah, great. Perfect. So, first of all, I would love to thank to NEMIS Technologies for giving us opportunity to bring on a streaming platforms such amazing experts in food safety, microbiologists, and really the best professionals in this field to share their knowledge. And today, I'm also super honored to welcome here my colleague, Dr. Julian Ihssen, definitely distinguished microbiologist and biotechnologist with over 25 years of experience. And that's not joke, because believe me, I'm working with this person every day, and his work has really significantly impacted both academic research and industry practices. And I would just ask you all to be active during the webinar session and type your questions in the chat room. It will be definitely answered afterwards, because our webinar topic today is about Escherichia coli, a hygiene indicator underappreciated in food industry. So, Julian, you are welcome to share about the topic, and the time is yours.
Okay, thanks a lot, Alina, for the very kind introduction, and welcome everybody to today's seminar. I'm going to start sharing my slides now. I hope everybody is seeing them. I also go to full screen mode. So today I want to talk about E. coli. And my a little bit provocative title maybe is Escherichia coli a hygiene indicator underappreciated maybe in food industry? I'm going to start now with a just very basic overview, some basic facts of E. coli. Many of you, I'm sure, know most of the facts that I present. It's already a long time that it was discovered by a children's doctor called Theodor Escherich, a German, who isolated this bacterium for the first time from newly born children, from neonates. It's a typical, I would say it's really a classical gram-negative rod-shaped bacterium. Gram-negative means, as you probably know, you have these two classes, broad classes of microorganisms of bacteria, the gram-negative and the gram-positive bacteria, which have a fundamental difference in their cell wall and membrane structure. And the gram-negatives, they have these two membranes and the periplasmic space in between. E. coli also is mobile. It has these nice flagella all around the cell, which it can then use to propel itself in specific directions. Also, it is a facultative anaerobe, means it can do oxidative metabolism, respire things with oxygen, but it can also do very well without oxygen and do a so-called mixed acid fermentation. That's what it does in its primary habitat, which I show just in a second what the primary habitat is. E. coli is really very well growing at 37 degrees. That's the optimal temperature, but it can grow up to 44 degrees. It's not so good at growing at lower temperature. And that's also important for its use as hygiene indicator, which I explain later. It's very fast growing. It can duplicate in as little as 21 minutes. And the ones of you which are familiar with the exponential growth curve may know what that means, how many cells there can be in some hours. So another feature of these microorganisms is that it is a very elaborate adaptation to stress and starvation. It's very flexible, stress resistant, and can also survive for a long time without any food. Also, for later, for the detection method, it's important that you have two very characteristic enzymes, which more or less all strains of these organisms harbor. It's beta-galactosidase, used for breaking down lactose, and beta-glucuronidase. And maybe a bit more fun fact is that it's really, I would say, the favorite lab organisms of the microbiologists, and it's used since 100 years in the lab, and a lot of the basic biochemical things and especially molecular biology mechanisms and gene regulation and all kinds of things has been studied with this organism. Also, it was the first organism on Earth for which the whole genome was sequenced already in the beginning of the 90s. I could not resist but to just show a slide from my PhD defense talk. Because I worked in my PhD thesis exclusively with E. coli, and the topic was on the adaptation of this organism to nutrient limitation to slow growth. And you see here how versatile E. coli is. You have this standard medium, the broth, which many of you also may know, it's called LB broth, also known since the 60s. And it's very good to grow this strain, this bacterium. And if they are very fast growing at the beginning of the culture, they can only use very few carbon and energy sources, nutrients, especially what they like most is just glucose. But then if you just feed the same broth, the same medium, but you post them under limitation, and you can do that in a bioreactor, in a continuous culture, and then they will have a steady state kind of growth. And in this case, it's really 20 times slower. But you see what they do now, they just take up everything what's there. They open up their whole broad catabolic and nutrient breakdown capacity. And these cells have also been called hungry and not completely starved. So it's kind of an in-between state. And that shows just how flexible this bacterium is. So the primary site where it lives is us. Us and other mammals, like cattle, pigs, dogs, mice, rats, all kinds of animals, but also birds, poultry. It always lives there, and it has been even studied in humans or also in mice where it's exactly occurring, and it's just at the beginning of the large intestine. As you might know, the large intestine is a gigantic, really very active bioreactor. And the E. coli is part of it, but it just grows right at the beginning. Some people say that its role is to use up all remaining oxygen, so that all these fermentative organisms in the large gut can then do their fermentation. So we have in our gut 10 to the 5 to 10 to the 8 cells of E. coli per gram. That's really not a lot, because the total number of living microorganisms in our gut is usually 10 to the 11. So E. coli is just 1% or 0.1%. What's also maybe important, it can also be carried around for houseflies. So there's really lots of study on these organisms, and they've really demonstrated that it's there on the flies' feet and so on. And that's another reason why it's such a a good hygiene indicator. So another important fact about E. coli is that you have both not dangerous commensal strains and also numerous pathogenic strains. So we all carry these microorganisms, but it does not cause harm. Some studies even say that it may be beneficial because it may prevent colonization of the gut wall with dangerous organisms. But it's not exactly known, but it's clear that it's a co-evolving bacterium, co-evolving with mammals and with us since many million years. So the pathogenic strains of E. coli, they have a lot of these abbreviations, which you probably also have come across already. So the general name for the ones which cause diarrhea and other diseases of the gastrointestinal tract, they're called EPEC, enteropathogenic E. coli. And then there's subgroups of this EPEC, for example, the enterotoxigenic E. coli, which produce certain toxins, which can lead to food poisoning. Then there's the enteroaggregative E. coli, which have a specific phenotype of disease formation where they do some aggregation of cells lining the gut wall. Then there's enteroinvasive E. coli. They're even more dangerous, and the most dangerous, really dangerous ones are the enterohemorrhagic E. coli, the famous 0157H7 serotype, also involved in litigation in the 90s and 2000s, as Bill Maher has explained in our last webinar. So this one causes bloody diarrhea, and in the worst case, it can lead to hemolytic uremic syndrome, so breakdown of the kidney. And all of these, if it's getting really worse, can also lead to sepsis. And then there's, on the other hand, the UPEC, the uropathogenic E. coli, which cause inflammation of the kidney and bladder. They're also quite a high disease burden worldwide, and around 90% of urinary tract infections are thought to be caused by them. And on top of that, there's a problem which is getting worse and worse. That's an antibiotic resistance. So it's really not usual anymore that they're resistant to simple antibiotics like ampicillin. But nowadays you find often strains which are also resistant to a whole class of antibiotics, like the extended spectrum bacterolactamases and also carbapinamases. Strains which harbor this enzyme, they're resistant to almost all known antibiotics. So that's really a big problem. The transmission of all these pathogenic E. coli is always via the fecal oral route, and that can then, in the end, also include food if the food is contaminated by feces. So what are at the moment the most common hygiene indicators in the food industry? So in Europe, it's really the most important ones is the Enterobacteria C, and that's really a large group of organisms, 30 genera, 100 species, including Citrobacter, Klebsiella, and all kinds of other species, which also occur in soil and fresh water and everywhere. So they're not really gut-related organisms. And this standard method is a quite simple method. It's violet red bile glucose agar. That the main things in there is that it's having bile salts and violet red, and suppresses by this the gram positives, and then it detects production of acid from glucose. But glucose, I mean, that's just used by almost all microorganisms, and that means that this is really you will detect a very large diversity of gram-negative bacteria. Then in USA, they have a different tradition, and there it's more the coliform bacteria, which are very important. And there's some classical methods like the McConkey agar, but also other well-known broth and agar medium. and very often a membrane filtration is done for the water analysis, and then you have chromogenic agars where you can detect these bacteria. And here for the, it's a bit more specific, the coliform test, because it's focusing on lactose, so the beta galactose positive bacteria. And it's important to know that E. coli is just like a subgroup of both of these two tests, so it can always be part of them. It's positive both positive both on VRBG agar and on the McConkey agar. So why, in my opinion, is E. coli a really good hygiene indicator? So first, humans, livestock, birds, pets, and so on, they all constantly shed E. coli with their feces because they are permanently colonized by it. But that's important. It does not occur just in the environment like that. It does not multiply there. It has no niches outside of the host. There's some exception that it can also multiply in food, for example, in milk. But I would say that's rather the exception, especially if the temperatures are not so high. And with respect to being an indicator, it's really a very good direct indicator for fecal contamination. And that means it may occur with other pathogens like enteropathogenic viruses, pathogenic bacteria transmitted by a fecal oral route. So, for example, norovirus or Campylobacter jejuni. And that's why it is a good risk indicator. Another thing why it's a good indicator is that it's very easy to grow. As I've already mentioned, it's very fast growing. That means that you have a relatively short time to results, and you don't have that much overgrowth problem like for other bacteria, because there's not that many bacteria which can outcompete E. coli if it has the good growth conditions and the right medium. So, and because of this, because it's so easy to grow, and we all have a lot of simple, cheap methods with high specificity available. And here comes into play the other enzyme that I mentioned, the beta-glucuronidase. This enzyme can break down mucus, and it's probably needed by E. coli in its normal habitat. And almost all strains of E. coli have that enzyme. And it's very rarely occurring in any other bacterial species. So, and that's why also you have this statement already in 1985 that E. coli from the Committee on Food Protection of the NRC in USA, that E. coli is the best indicator of fecal contamination among the commonly used fecal indicator organisms, and it's the best indicator associated with hazard. So it's, as I mentioned already, used since a long time, and especially important for the water sector, drinking water, source water, but also recreational waters. Here, the standard is usually in a certain amount of water. E. coli must not be detected, especially if it's used for drinking. So the history of methods, so since a long time, the coliform test is known. McConkey was the one who really invented the method. At the beginning, it was just gas production from lactose, but then later on acid production, giving some pH indicator, color change. Then this method was modified and improved by increasing the incubation temperature to 44 degrees. That was then called so-called fecal coliforms, and that it's a bit more selective for E. coli. And since the 70s, we have synthetic chromogenic and fluorogenic substrates, also always for beta-glucuronidase. And that's still very popular today to have chromogenic media, which give then blue or red colored colonies on agar plates if you have E. coli. Then certainly since the 90s, a lot of different molecular methods are available, PCR later on, also the real-time PCR immunoassays. And also since the 19th, we have combined coliform E. coli chromogenic agar plates, which have two different color-generating substances in them. And based on these substrates, there's also products like the Petri films you may be familiar with, and also dip slides. Really a lot of methods available. And since the 2000s, there's a newer thing we have is the synthetic luminogenic substrates, and that's exactly what NEMIS uses, and I will explain in a minute. So what are the requirements by law? So there is certain regulations for E. coli, especially very important, the one for drinking water. That's, I think in most countries, it's E. coli must not be present in 100 milliliter drinking water. So, but also in the food sector, we have regulations, and seafood, finished seafood products. It should not be 1 mpn, that's another method, but it also means 1 cell per gram and maximum tolerated 10, if not all samples are positives. In living seafood, it can be higher, but there really you have always a risk of this virus transmission because they take up these pathogens from the seawater, which is contaminated with wastewater. Then you have the minced meat and separated meat should be below 50 CFU and so on. Also, there's some regulation for ready-to-eat fruit and vegetables, shouldn't be above 100 CFU per gram, but also for cheese, which has been made from heat-treated milk, it really should not be more than 100 CFU. So it is already some requirements by law, but as far as we know, as NEMIS, when we talk to customers, is that rarely anybody does something like environmental monitoring for E. coli. As I mentioned already, it can be that E. coli grows somewhat in food. So there's, for example, a study from 2018 where they show that EHEC increases from 200 to 600 CFU in gram in 1 day and then stays constant during the ripening process for such a ricotta type goat cheese, and it's still detected in the final product after 90 days. And then there's also, for example, some simulations which have been calculated in a computer program, theoretically how many E. coli can be there after 1 day, and it's quite substantial in milk that they can grow fast. So for our test, we use exactly the same principle as used for this chromogenic agar plates. There is a substrate for beta-glucuronidase, and it's but in this case, in our case, it's attached to a so-called dioxetane molecule, which will emit light. It is chemiluminescence once it is cleaved, and that we detect then finally with our reader, and that's a very sensitive method to detect this activity. Our test procedure is quite straightforward. So take the sample, wet it in a wetting solution, then put it in the tube. You close the tube. We have a biosafety tube, so it's really safe afterwards, incubated, and it's for E. coli, it's really enough to do 16 to 18 hours, and we've shown that really 1 CFU of dried cells can be detected after that time. If you have higher amount of E. coli, you can even reduce the incubation time to 6 to 8 hours. Then after this incubation, you press the button of our cap and release the tablet with the luminogenic substrate, with the AquaSpark substrate, and read the results. And we have set a certain threshold once it is positive for E. coli. There can be confirmation with the PCR or qPCR afterwards directly from the broth. Here to show you that it's a quite robust test. So inclusivity, we have tested more than 100 strains from our strain collections, and all of these isolates are from either from food, from source water, but also clinical isolates, and strains from well-known strain collections like ATCC. And you see here almost all of them are positive, and all of them give a really high signal. So the threshold is 10,000, and they go up to 6 million and more, most of them, meaning that you have more than 100-fold signal to background ratio. Also, with respect to specificity, as I already mentioned, there's very rare other bacteria which have this enzyme. And as expected, then all kinds of different species, Bacillus, Proteus, Serratia, which occur a lot in the environment and in food factories, Enterococcus, Aeromones, and so on. All of these are clearly negative in our test. So then to prove that more like application-oriented situation is also covered by our test. We tried cells, E. coli cells on stainless steel. And also here, we can go down to very low numbers of inoculated cells, but you have to bear in mind that only like 4 to 13% of the cells survive. And also we find almost all tests clearly or strongly positive after our recommended incubation time of 16 to 18 hours. So also, finally, we tested if we can also artificially contaminate food. And also this had quite some good results when we put E. coli in very low numbers on food surfaces, let them sit there for 3 hours. We can readily detect it with our test reliably and reproducibly, down to also single-digit numbers of inoculated bacteria or viable bacteria. So finally, also our test might be useful for water samples, just added, for example, with a single-use Pasteur pipette, tried different volumes from 20 microliter to 1 milliliter, which would be 1/3 volume to the broth volume we have in our tests. And here also they were all reproducibly positive when we spiked just normal tap water. So luckily our tap water did not harbor any E. coli, as you see. But if you spike them with a low number, a single-digit number of cells, then clearly also positive with a signal-to-background ratio of more than 100. Okay, so just for summary, because we're already out at the end of the time which I have for this talk. So the summary is that E. coli, in my opinion, is a very useful hygiene indicator also in the food industry. It specifically indicates the risk of fecal contamination, so it specifically indicates high hazard. And then you have a lot of different, numerous, well-established, simple-to-use E. coli tests that you can choose from for monitoring your hygiene status. And we think that we offer quite a good solution to detect this organism with our new luminogenic swab test. The test is lab-free, it's safe because you close the tube with the biosafety cap, and you can only open it with a tube cutter afterwards. It's robust, as I've showed in our application data. It's relatively fast, and it can be easily be done by any factory worker after a short training. So with this, I'm at the end of my presentation, and I'm very happy to answer any questions you have regarding E. coli. Thanks for your participation.
Thank you very much, Julian. And definitely, we already have some questions. So people were actively listening and curious to know a lot of stuff. So we start with the first one from Ismail. The question is, from European and US hygiene indicators, which indicator will you suggest? Any justification? Many thanks.
So, mm, well, from the regulation, they're sometimes required to do coliform tests, and in Switzerland, enterobacterial tests for finished products, for final products. So that's, in my opinion, yeah, you just have to do it, and it's also makes sense in often in many cases. But it could be that for monitoring your risk inside the factory, and also something like hand hygiene and handles and so on, it could make sense to also do some E. coli testing. And as I said, I don't know about other countries than Switzerland, but there are some regulations, especially for seafood, where you have to test also the final product for E. coli.
Okay, thank you, Julian. Next question comes from Angelica. E. coli requirements by law. There is a list of limits for food. What is one for dry products? For example, granola, oats.
I'm not aware that there's any regulation for dry food. There, I cannot tell really. One would have to go and search. Probably there's none.
Okay. Next question comes again from Ismail. In requirements of laws, should E. coli count be enough for safety insurance despite some E. coli are stag strains?
The insurance, yeah. So I think if you make sure that you bring down your number of positive E. coli tests, or ideally don't find any E. coli in your factory, especially in the high risk zones, then you can be pretty sure that the likelihood of having STEC is low. And many of the pathogenic strains will also be positive in the glucuronidase-based methods. But EHEC is one of the few which are not glucuronidase positive. But still, from the risk point of view, it might be better to check for E. coli because they're much more often occurring, and EHEC will be very, very rare. And to reduce the risk, to monitor E. coli, I think, is maybe a good idea.
Okay, thank you, Julian. Next question comes from Rainer. Which food surfaces did you test as well, besides mentioned ones? Any list available?
We just tested these three, but we are very welcome for any suggestions you might be interested. We are also very open to collaborations with companies, and then we can do some tests in our labs. Yeah, but I think it's because it's quite robust. As long as you don't introduce too much food residue into the test, it should work. But yeah, we have to, in the end, test it for every different food.
Yeah, there is a clarification from Rainer also. What about meat carcasses?
It should work, but again, maybe it would then be good to compare it to other methods also, like classical plating, send it to a lab. But probably it will work.
Okay, next question comes from Angelica. One of our products has constant presumptive E. coli results, which is then not detected after retest. Is there anything that could cause false positive? Could it be caused by product type? It is always presumptive and never came back as detected after the retest.
It could be that there's some bacteria in specific settings on specific foods which also have this enzyme. That's the one thing. And I don't know, maybe there's also some food which may have this activity, but here we don't have that much experience yet, but it's also something one could investigate in the lab. And also here we are willing to do some tests if we can buy such food from supermarket and just check if we get false positive results, and then if we can spike it with E. coli. Yeah, the test is very new, so we don't have extensive experience yet with it. But we are very happy anybody wanting to try it out, and we can also discuss if you encounter some problems.
Until our participants type in more questions, because we also have currently live streaming our presentation in LinkedIn. So I'm just really encouraging the one who watch us on LinkedIn to type the questions in the comment. So I would ask one more question from my side. Have we done any studies with our N-Light test in food factories?
Yeah, we've done once a so-called "Send Your Sample" campaign. So we sent out the test for free, and people took some samples and sent it back to us. There we got like 1 to 10% positive samples. So in principle, it worked, and we also could confirm in some of them presence of E. coli with PCR. And it's not that ideal that you send the sample overnight. It's better to really start incubation quite soon, but nevertheless, it seemed to work in this case. And this was cheese factories, it was fish, like smoked salmon factory, and meat factories. So in these three sectors, in principle, it seems to work. Yeah.
Okay, and what about agar plates? Can you also confirm that it works? I mean,
Yeah, that's a bit a hassle, because this enzyme which we detect is inside the E. coli cells. So we have developed a specific lysis method, which we add these agents we add with the tablet, and therefore the cells are destroyed, and you cannot subculture them. So but it is possible to do a confirmation by PCR. We've done that already with some published primers specific for E. coli, and it works quite well. But subculturing in this case is not possible. But anyway, it's more really intended as an hygiene indication test.
Yeah, and at the end, to finish the topic about our test, how long is the shelf life of our test?
Oh, it's a very robust test. It's the shelf life is 12 months. We are now thinking even of increasing it to 50 months, because this chemiluminescent substrate that we use is very stable, and the broth also is quite simple and very stable. So here, yeah, it's really quite a robust test.
Okay, so if there is no more other questions, then I would just thank to all of you guys who participated today. Thank you for your questions today. If someone want to ask something, you can still type it. I just want to announce our next webinar. Next webinar is gonna be 11th of June, so save this date in your calendar, and it will be also something that you should not miss. We will talk about crisis management and incident handling in the food and beverage sector. And the expert we are waiting for is Professor Bijan Purkamalian. He is also microbiologist. He has PhD in microbial psychology, and also he's currently the general manager in McDonald's. So it's something that you should not miss out, definitely. And biggest thanks to you, Julian, for giving your time to us, to sharing your experience.
Welcome. Very welcome.
Yeah, happy to see you for the further webinars. And guys, see you next time. Have a nice day. Bye.
Thank you all for attending, and bye-bye.